we'll begin with glycolysis. Okay, sorry, we'll begin gluconeogenesis. Okay, so gluconeogenesis, this is the process of synthesis of glucose from non-carbohydrate source, and in mammals some tissues depend almost completely on glucose for their metabolic energy. For the human brain, nervous system, as well as kidney, medulla, erythrocyte, testis, and embryonic tissues, glucose from the blood is the sole or major fuel source, and the brain itself alone requires about 120 grams of glucose per day. That is more than half of all the glucose stored as glycogen in the muscle and liver. However, the supply of glucose from these stores is not always sufficient between the meals and during longer fast or after vigorous exercise, so glycogen is depleted. For this time, organisms need a method for synthesizing glucose from non-carbohydrate precursor. So this is accomplished by a pathway called gluconeogenesis, formation of a new sugar. And gluconeogenesis pathway converts pyruvate and related 3 and 4 compounds to glucose. So there are some facts about gluconeogenesis. In many microorganisms, gluconeogenesis starts from simple organic compounds of 2 or 3 carbons, such as acetate, lactate, or propenoid in their growth medium. Although the reaction of gluconeogenesis are the same in all organisms, the metabolic context and the regulation of the pathway may differ from one species to another and from tissues to tissues. In plant seedlings, stored fat and proteins are converted via paths that include gluconeogenesis to the disaccharide sucrose for transport throughout the developing plant. And this is the transporting form of carbohydrate in plant. And glucose and its derivatives are precursor for synthesis of plant cell wall, nucleotides, coenzymes, and a variety of other essential metabolites. So in photosynthetic organisms, they use this pathway to convert the primary product of photosynthesis into glucose to be stored as sucrose or starch. So this is the overview of gluconeogenesis. So it occurs in all mammals, in animals, in plants, fungi, and microorganisms. The reactions are essentially the same in all tissues and species. The important precursor of glucose in animals are 3 carbon compounds like lactate or pyruvate or glycerol, as well as certain amino acids. In mammals, gluconeogenesis takes place in the liver and to a lesser extent in renal cortex and in the epithelial cell that line the inside the small intestine. The glucose produced passes into the blood to supply other tissues. After vigorous exercise, lactate produced by anaerobic glycolysis in skeletal muscle returns to liver and is converted to glucose. So here, if you begin with lactate, it will be converted to pyruvate, then enter into the TCA cycle to produce phosphoryl pyruvate, and phosphoryl pyruvate to glucose 6-phosphate through different sequential reactions, and glucose 6-phosphate to finally glucose. Then they can contribute to the blood glucose level increase, glycogen synthesis, glycoprotein synthesis, disaccharide synthesis, other monosaccharide synthesis, starch synthesis, or sugars. And there are some glucogenic amino acids. The amino acids that contribute the production of glucose is called glucogenic amino acid. They can enter into the citric acid cycle, convert it to phosphoryl pyruvate, then phosphate, glucose 6-phosphate. And lipids or fats are converted to glycerol. Then this glycerol can also enter there to produce glucose 6-phosphate. So these are 3 important mechanisms in animals. And what about the plants? They start from carbohydrate fixation to produce 3 carbon compound 3-phosphoglycerate, and then this contributed to their producing glucose 6-phosphate. This is how they can contribute to the production of glucose through gluconeogenesis. So gluconeogenesis versus glycolysis. Gluconeogenesis and glycolysis are not identical pathway running in the opposite direction. Although they do not share, although they do share several steps, 7 of the 10 enzymatic reactions of gluconeogenesis are the reverse of glycolytic reactions. However, 3 reactions of glycolysis are essentially irreversible in vivo and cannot be used in gluconeogenesis. Those are the conversion of glucose to glucose 6-phosphate by hexokinase. This is a irreversible reactions, and the phosphorylation of fructose 6-phosphate to fructose 1,6-phosphate by phosphofructokinase 1, and the conversion of phosphoenol pyruvate to pyruvate by pyruvate kinase. So these are 3 important reactions that are irreversible in glycolysis, and this is and through this enzyme we can control the glycolysis. So here you can see the process of glycolysis and the process of gluconeogenesis. So from upper to lower is called glycolysis, and from bottom to upper is gluconeogenesis. So here we can see the conversion of, if you consider the gluconeogenesis, the first is the conversion of pyruvate to phosphoenolpyruvate. So this occurs in two steps. In the first step, pyruvate is converted to oxaloacetate through the enzyme pyruvate carboxylase, and then oxaloacetate will be converted to phosphoenolpyruvate. But this conversion occurs in cytosol. So what happened? Oxaloacetate needs to cross through the mitochondrial membrane to the cytosol, because oxaloacetate is synthesized in the mitochondrial matrix. So what happened there, when oxaloacetate is synthesized in mitochondria, they will be converted to malate. They will first convert it to malate through malate dehydrogenase enzyme, and then malate will be crossing the mitochondrial membrane, because the mitochondrial membrane does not contain any transporter for oxaloacetate, and before export to cytosol, my oxaloacetate form from pyruvate must be reduced to malate, and then malate will goes to the cytosol, and in the cytosol the malate will reoxidize to oxaloacetate with the production of cytosolic NADH. So this is two-step process. So first one, pyruvate is converted to oxaloacetate by pyruvate carboxylase. This occurs in mitochondrial matrix, and oxaloacetate cannot cross the mitochondrial membrane. So oxaloacetate has to be converted to malate, and malate transfer through the mitochondrial membrane to the cytosol, and in the cytosol malate converted to oxaloacetate again, and oxaloacetate to phosphoenolpyruvate occurs in the cytosol by phosphoenolcarboxylase, because this enzyme located in the cytosol. So synthesis of phosphoenolpyruvate from pyruvate. So the similar reaction that has been shown here. So pyruvate combined with bicarbonate to produce oxaloacetate. So glucose biosynthesis cannot proceed unless NADH is available. The transport of malate from mitochondria to cytosol and its reconversion in the cytosol to oxaloacetate effectively moves reducing equivalent like NADH to the cytosol. This is how they maintain their concentration. And this path from pyruvate to phosphoenolpyruvate therefore provides an important balance between NADH produced and consumed in the cytosol during gluconeogenesis. So what are the alternative pathways? So we have already studied this one. The pyruvate will enter into the mitochondrial matrix, convert it to oxaloacetate, then malate will goes out through the mitochondrial membrane to the cytosol, and malate to oxaloacetate and phosphoenolpyruvate. So there is another pathway when lactate the concentration of lactate is higher. So this is the alternative pathway, and this pathway activated when the concentration of lactate is higher, or when the demand of NADH is higher, or the concentration of NAD plus is higher. So in this way, lactate converted to pyruvate through the production of NADH, then pyruvate will enter into the mitochondrial matrix, will be converted to oxaloacetate, then converted to phosphoenolpyruvate. This is the mitochondrial phosphoenolpyruvate carboxykinase. Then this phosphoenolpyruvate enters to the cytosol and contribute to the gluconeogenesis pathway. So the bypass reaction in gluconeogenesis, so conversion of fructose 1,6-bisphosphate into fructose 6-phosphate. This is the second. So already here phosphoenolpyruvate is formed, then phosphoenolpyruvate will convert it to fructose 1,6-phosphate through different sequential reactions, and now fructose 1,6-bisphosphate to fructose 6-phosphate. In this reaction, fructose 1,6-bisphosphate is converted to fructose 6-phosphate by fructose 1,6-bisphosphatase. That means removal of phosphate group and producing fructose 6-phosphate. And then finally, glucose 6-phosphate will fructose 6-phosphate then converted to glucose 6-phosphate here, fructose 6-phosphate to glucose 6-phosphate, and glucose 6-phosphate is finally converted to glucose through glucose 6-phosphatase. This is the phosphatase enzyme that can trim the phosphate group. This magnesium activated enzyme is found in the luminal site of endoplasmic reticulum of hepatocyte and renal cell. Muscles and brain tissues do not contain these enzymes, and so cannot carry gluconeogenesis. And glucose produced by gluconeogenesis in the liver and kidney or ingested in the diet is delivered to brain and muscle through bloodstream. So glycolysis and gluconeogenesis are regulated reciprocally. So at the 3 points where glycolytic reactions is bypassed by a different type of enzymatic reaction in gluconeogenesis, simultaneous operation of both pathways would be wasteful. So if you consider the glycolysis goes through this way and gluconeogenesis, the glycolysis goes through this way and gluconeogenesis goes through this way, so this will not ultimately produce any glucose. This is just wasting of energy. So that is said here, if glycolysis, the conversion of glucose to pyruvate, and gluconeogenesis, the conversion of pyruvate to glucose, were allowed to process simultaneously at higher rate, the result would be the consumption of ATP and the production of heat. For example, phosphofructokinase 1 and phosphate fructose 1,6 bisphosphatase catalyze opposing reactions. So to sum up this, if you sum up this reaction, you can see this one. So the sum of these reactions is an energy wasting reactions resulting in hydrolysis of ATP and production of heat without any net metabolic work being done. So what are the 2 alternative fates of pyruvate in aerobic respiration? So pyruvate basically converted to acetyl-CoA and citric acid cycle, and acetyl-CoA enters into the citric acid cycle and then energy. So if the concentration of acetyl-CoA is increased, that means your body has a loss of energy. So acetyl-CoA will stop pyruvate dehydrogenase complex. That means it will stop the conversion of pyruvate to acetyl-CoA. So with that, acetyl-CoA also accelerate the conversion of pyruvate to oxaloacetate by pyruvate carboxylase. So then this oxaloacetate contribute to the production of glucose through gluconeogenesis. So these are the sum of gluconeogenesis and glycolysis. So you can see the gluconeogenesis is a energy consuming process than glycolysis. So here you can say in glycolysis 2 ATPs are synthesized, but in gluconeogenesis 4 ATPs, 2 GTPs are consumed. So let's study the pentose phosphate pathway. It's a very important pathway. So we know 90 to 96% glucose after diet goes to the glycolytic pathway. So the rest of the 4 to 6% of glucose, depending on the situation, will enter into the pentose phosphate pathway. So pentose phosphate pathway has 2 phases. One is non-oxidative phase and oxidative phase. Those 2 phases depend on the demand of final product of each phase. So if the final product, if the demand for ribose 5-phosphate is higher, then oxidative phase will be conducted, or if the demand of NADPH is higher, then oxidative phase will stop. So ribulose 2 glucose 6-phosphate and NADPH is produced. So this is non-oxidative phase. So in pentose phosphate pathway, NADP is the electron acceptor and yielding NADH, and rapidly dividing cell such as bone marrows, skin, intestinal mucosa use the pentose phosphate pathway to make DNA, RNA, nucleotides, ATPs, NADH, coenzymes. But in other tissues, the essential product is not ribose, but the electron acceptor NADPH, and this is needed for reductive biosynthesis or to counter the damaging effect of oxygen free radicals. So if the concentration of NADPH is higher, then the body is in relaxed stage, or if the concentration of NADPH higher, then it shows your body is oxidative stress. So NADPH will convert the oxidized glutathione to reduced glutathione by glutathione reductase, and also NADPH can be used for biosynthetic precursor catalyzed NADPH catalyze the reductive biosynthesis reaction, production of fatty acid, cholesterol, sterols from precursor molecules. So tissues that carry out extensive fatty acid synthesis or very active synthesis of cholesterol and steroid hormones require NADPH provided by this pathway. By maintaining and reducing atmosphere a high ratio of NADPH to NADP and a high ratio of reduced to oxidized glutathione, they can prevent or undo oxidative damage to protein, lipids, and other sensitive molecules. Erythrocytes and the cell of the lens and cornea are directly exposed to oxygen and thus to damaging free radicals generated by oxygen. In erythrocyte, the NADPH produced by pentose phosphate pathway is so important in preventing oxidative damage that a genetic defects in glucose 6-phosphate dehydrogenase, the first enzyme of the pathway, can have serious medical consequence. So, glucose 6-phosphate is partitioned between glycolysis and the pentose phosphate pathway. So, glucose to glucose 6-phosphate formation is same in glycolysis and pentose phosphate pathway. So, when glucose 6-phosphate is converted to when the concentration of glucose 6-phosphate is higher, it will either can goes to glycolysis or can enter into the pentose phosphate pathway. But it depends on the needs. So if the demand for pentose phosphate or if the demand for NADH is higher, so it might enter into the pentose phosphate pathway rather than entering glycolysis. Of course, glycolysis will be continuing, but the ratios or the quantity will be slightly increased. So whether glucose 6-phosphate enter glycolysis or pentose phosphate pathway depends on the current needs of the cell and on the conversion of NADP in the cytosol. When the cell is rapidly converting NADPH to NAD in the biosynthetic reactions, the level of NADP will be raised allosterically, stimulating the glucose 6-phosphate and thereby increasing the flux of glucose 6-phosphate through the pentose phosphate pathway. So that's all from here in this lecture.